In the first lecture, I mentioned just briefly a little bit about susceptibility. But susceptibility to autoimmune diseases, although more common in females by far, it does seem to run in families. And it does vary among different populations or ethnic groups. And so there could be some sort of genetic influence involved. Um, and genetic factors do confer differential susceptibility to autoimmune diseases. So individuals like rheumatoid arthritis, for example, we see that run in families. And so there's probably a genetic basis behind that. And some of the most important genetic factors that influence susceptibility are in those HLA genes, those HLA, the human leukocyte antigen, that complex of genes. And that makes sense because that's what we see with the immune system as being those self-antigens. And so almost every autoimmune disease that we know has some sort of HLA association. And we do know that autoimmune diseases are most prevalent in Caucasians than any other population. And they are much more prevalent in females than they are in males. So this graph here on the side shows some of the major autoimmune diseases that you've probably heard about, and they were included in those tables a few slides ago and in your text. But you can see that females by far have the most what's the word I'm looking for here? Prevalence of autoimmune diseases. The only autoimmune disease that is more commonly found in men than it is in females is ankylosing spondylitis. And otherwise, all of the rest of them are either more common in females or about the same. So that's kind of interesting. So hormones might be involved as well as genetic differences. So the functions of these polymorphic HLA class 1 and class 2, remember, that's the same as MHC class 1, class 2. HLA and MHC are the same. It's just we talk about HLA in this context. They are going to be the ones that are going to present the peptides, right? Recall MHC class 1 will present to CD8 positive T cells, where MHC class 2 will present to CD4 positive T cells. So although some autoimmune diseases are mediated by antibodies, you have to remember that antibodies really don't come about unless they have T cell help, right? So they can be an antibody-mediated disease, but even at that, the T cell that is the B cell soulmate also had to lose tolerance as well, or breach tolerance. And so we see that T cells are going to be involved, whether it's antibody mediated or T cell mediated. So the number, the nature, the strength of the disease then is going to really depend on that loss of T cell tolerance to some self-antigen. And then that is the first necessary step to development of any autoimmune disease. And then the fact that an HLA allotypes are associated with disease does suggest that the first step includes some sort of presentation of that self-peptide bound to one of those MHC molecules, has to be recognized by that clone of T cells that somehow breach tolerance. So there's kind of a lot of things that have to occur in order for an autoimmune disease to fully develop. So the disease association with HLA are also very consistent with T cells being more important for preventing autoimmunity than B cells. Because if a T cell is fully prevented from recognizing self-antigen, it's not going to get out into circulation. It will be selected through negative selection and removed, or made anergic, or gone back through receptor editing, to change its T cell receptor specificity. And then that would prevent a soulmate T cell to find a soulmate B cell with the same specificity, the self-antigen. And so really autoimmunity does rely on the loss of T cell tolerance. And maybe you recall that when we talked about the development of T cells in comparison to the development of B cells, there was a much more stringent negative selection on T cells than there were on B cells. those mechanisms for removing autoreactive cells from that naive B cell population are not as good. So the bone marrow is okay, but it's pretty much restricted to the peptides that are presented in the bone marrow. But recall that in the thymus, those thymic stromal cells had that error gene, which provided the ability to those cells to transcribe and translate self-peptide from peripheral tissues. And so that thymus just had a wider range of self-peptide to test these T cells with. So quite honestly, there's many more circulating B cells that have specificity to self-antigen than there are T cells. It's just the B cells never get activated because they never find their soulmate. So they're just going to be single for the rest of their lives out there, just searching, but never being able to find that T cell that can make it, because the T cell got hung up in the thymus and removed from circulation. I kind of joke and I make these analogies, but I think it helps kind of draw that picture or paint that picture for you that B cells are dependent on T cell actions in order to become fully activated. So many more autoimmune diseases are actually associated with HLA class 2, or in other words, MHC class 2, than those associated with MHC class 1. And so what that really kind of tells us is that a lot of autoimmune diseases are activated by CD4 positive T cells than CD8 positive T cells. And this propensity that we have for naive T cells then really kind of hangs with it. There's a lower threshold than there are for naive CD8 positive T cells being activated by foreign antigen. So CD4 positive T cells play a bigger role in autoimmunity than CD8. That's what that's saying. But yes, we do have autoimmune diseases that result because of antibody binding to a cell surface receptor. When an antibody binds to a cell surface receptor, it actually mimics the ligand for that receptor, and any signaling that occurs or that is supposed to occur when that receptor is initiated or bound, that's going to happen. And so autoantibodies that are receptor agonists mimic that natural ligand of the receptor and cause the transduction of signals to occur just as if the actual ligand had bound. Even though the ligand had been bound, it was an autoantibody. Now, autoantibodies that are receptor antagonists don't activate the signaling on binding. They actually block the actual or the natural ligand for that receptor, and then they prevent signaling. So you can have agonists and you can have antagonists, and that's pretty consistent with other terminology and other courses that you've probably learned about. It's just we have autoantibodies that are binding up those receptors and either acting like the ligand in an agonist case or acting as a block to the ligand in an antagonist case. So we use this idea of antagonists gosh, go back here. We use this idea of agonists and antagonists, specifically antagonists, when we look at therapies for autoimmunity, like IVIG or monoclonal antibody treatments. And they are going to, the idea behind using a monoclonal antibody would be to saturate the binding sites of the autoantibody for IgG, those Fc gamma receptors, and preventing the actual autoantibodies from binding. So they're agonists or antagonists, sorry. And then this works pretty well. The problem with this is that the patient usually is not going to recognize they have any autoimmune disease until it's pretty far down the line. And the destruction of the target tissue is to the point where symptoms have started to appear. And by that time, it's hard to go back. So if you start having joint damage because of a rheumatoid arthritis or a lupus, by the time somebody feels the symptoms, the disease has progressed so far that it's tough to treat in a way to reverse what has already happened. So that's a big downfall with autoimmune diseases and treatment. And for almost every autoimmune disease, there are clinical observations suggesting that there might be some sort of true protective case that occurred where there really was an infection of some sort and there really was a true immune response, but it went haywire for some reason. And led to the development of an autoimmune disease. Now, this isn't set in stone. A lot of this evidence can be correlative, and we know that correlations do not mean causation. And if you don't know that, you should know that. Correlation, sure, you can correlate things, but it doesn't automatically mean that it's a cause. Sometimes it's just completely circumstantial where it's like, oh my God, like this just happened at the same time this happened. It doesn't mean that it caused it. And then sometimes it's just anecdotal where it's like, yeah, I heard about this one person, it's a story kind of. It doesn't mean it's not true. It's just not hard scientific evidence that we can use to support or refute a hypothesis. The fact that genetically predisposed individuals develop autoimmune diseases with environmental factors like smoking or physical trauma, could smoking lead to a higher? Maybe, but it's tough to say. And so really what it comes down to is a lot about what we know about how autoimmune diseases develop. We don't really know too much. It's all kind of just speculation or correlation or anecdotal, right? But what we do know is that maintaining T cell tolerance is central to prevention or treatment of autoimmune diseases. And the unanswered question here is how this self-tolerance is affected by gradual decay of the thymus. Recall a long time ago when we talked about T cell development, we said that the thymus is where T cells get educated. And what we meant by educated was that they learned the difference, or they learned to discriminate between self-antigen and not self-antigen, and they went through negative selection and then positive selection. But because the thymus doesn't hang around forever, and by the time we hit puberty, our thymus is pretty much a big fat pocket, it doesn't work forever. And so there is a reduction of the effectiveness of the thymus being able to educate new T cells that are developed. So by the time you're 50, if you don't even have much of a thymus at all, and you start developing T cells that have to go through negative selection, well, there's only 20% of the thymus left to do any of that negative selection. The chances of T cells slipping through that are recognizing self is a little bit higher than it was when an individual was 2 years old. And by the time a person hits 70, well, there's practically no thymus left to do any negative selection at all. So because our lifespan has increased so much in the last, 75 years, 50 years, we're living much beyond 70, and people that are 70, that's like the prime of their lives, right? And so we do see a lot more autoimmune diseases come up now than we did in the past because people didn't live as long, and then their thymus was, still working by the time they were hitting their old age. So by their nature, T cells populations are dynamic. T cells are going to divide periodically, and they're going to hang out for a long time, and they're constantly being replaced. And if that thymus isn't there to fulfill the needs of negative selection, there's a chance, pretty good chance, and it increases with age, that naive T cells that have specificity for self-antigen are going to get out there. And so then that can lead to a higher incidence of autoimmune diseases over a lifespan. You don't see autoimmunity too often in young children, type 1 diabetes being an exception. But mostly people get them a little bit later in their lives. So I gotta drink some coffee here. All right, now Tissue disrupting diseases, which do not involve antibodies or effector T cells, are characterized by chronic recurrent bouts of systemic inflammation. And we really have just been driving the fact home that adaptive immunity has been the primary and only cause of autoimmunity, but the innate immune system can cause increased inflammation, which can be destructive of tissues. And so distinguish these diseases from the autoimmune diseases, we call these autoinflammatory diseases. So they're not tissue specific. There's not recognition of specific cell antigen. It's just a non-specific inflammation that can lead to destruction of tissue. And so it's truly not an autoimmune disease, it's an autoinflammatory disease. But like autoimmune diseases, they still tend to run in families, and they tend to be more prevalent in populations, in certain populations over other populations. So there are some similarities. The big difference is that autoinflammatory diseases are with the innate immune system. Okay, so that kind of wraps up our tiny mini discussion on autoimmunity. And this topic, along with the previous two topics, remember the allergy and organ rejection, those are three situations in which the immune system does the job of the immune system, it just does it when it's not supposed to. And so even though the immune system thinks it's acting helpful, it's actually causing disease in that individual, and it's making the body unhealthy. And so this is really a case in which where the immune system is supposed to attack pathogens or cancerous cells that are unhealthy tissue. We have an instance now where we have allergy and we have transfusions or transplants, and we have autoimmune diseases where the immune system does its job, but it does it when it's not supposed to. And so those three situations all mimic these hypersensitivity reactions. And so this topic of the chronic autoimmune diseases caused by activation of T cells and B cells that recognize healthy human tissue, it falls in line with these hypersensitivities that we've been looking at the last now three topics. So although there's considerable clinical diversity in autoimmune diseases, allergic reactions, transplanted tissues, the underlying mechanism that crosses all three of these areas is limited to these four types of hypersensitivities. There's going to be IgG mediated, which we see with the allergies. There's going to be type 2, which is going to, IgE for type 1, right? IgG for type 2 and type 3, where we have these antibodies interacting. And then with type 4, we have cell mediated. So anyway, I think this is a really nice table to kind of just show how those hypersensitivity reactions kind of span through these three different situations where we have the immune system acting inappropriately. So, all right, that's the end of this topic, and we'll see you guys in class.